I'd just like to start by thanking the organizers for inviting me. It's such a pleasure to be here, and I've really enjoyed the talks from yesterday. I learned a lot. I hope you guys did too. I have to say, I was a little bit nervous when I got this invitation to speak because, number one, I'm not a clinician. I'm a cell biologist, and you might call me a geneticist, but definitely not a clinician. And number two, I was asked to speak on this subject of resistance to antifungals in Cryptococcus neoformans. And if you remember our talks from yesterday, or if you read any of the recommendations about Cryptococcus, clinical recommendations, what you'll know is that basically there is no resistance to antifungals in Cryptococcus, so how am I supposed to talk about it? So what I'd like to do in this talk is convince you that actually resistance to antifungals is a major problem in Cryptococcus. And also talk to you about a new morphotype that's been recently described called Titan cells and how that relates to those two, how that relates to this problem. But because of that, I'm going to spend quite a lot of time doing a review of the literature and some of the larger work that's been done that's not coming out of my lab, and then I'll conclude with the work that my lab is doing to try and tackle this problem. So is this? All right, so Cryptococcus neoformans. So you all know, we know that Aspergillus, all these beautiful movies, horrific movies yesterday of Aspergillus growing in the body and growing in an invasive way. We know that Candida albicans is a commensal that escapes the gut and grows as a hyphae and is invasive in organs, in tissues, like kidneys that we saw yesterday. Cryptococcus causes disease simply through proliferation. This is not an invasive organism. It's not hyphae that we worry about. It's the yeast. It's budding as a yeast. And what you can see here is just that action of proliferating, going from one cell to two cells. And this is what patients who have cryptococcosis are experiencing in their brain. So this is generally a disease where you inhale the spores, they translocate to the central nervous system and brain, and then cause meningitis. And that's mostly what that patients are presenting with. It particularly came to the fore in the 80s with the HIV epidemic. It was actually one of the indicative diseases that the AIDS epidemic was going on. And you can see here a striking picture from one of these first patients who was diagnosed with this disease, looking at histology of the brain after they succumbed to the infection in 1983. And what you can see in the brain quite nicely is yeast cryptococcal cells. And I think that's a really important thing to note, is that it's not the invasion of this organism that's the problem, it's the spread and proliferation of the yeast. I'll just give a quick overview of this problem. We all know these issues, but we estimate that there are 1 million Cryptococcus infections every year. Most of those are in sub-Saharan Africa, concomitant with the HIV/AIDS epidemic, but they do occur in other places. And then we estimate there's about 600,000 deaths every year, again, mostly in sub-Saharan Africa. But I would like to highlight that it's not simply HIV/AIDS patients that are suffering from this problem. So if you look at this cohort from Vietnam, what you can see is that about 29% of Cryptococcus patients are not HIV infected, and so you do need to be concerned about this organism even in the absence of an HIV infection. All right, so as I mentioned, if you look at the ISDA and the WHO guidelines for treating Cryptococcus neoformans, well, we're already ahead of the game here in terms of preventing drug resistance, because what they are recommending is combination therapy, right? So you do a 2-week induction of amphotericin B followed by flucytosine. In the absence of flucytosine, they're recommending a 2-week induction therapy again, plus fluconazole, and then long-term fluconazole maintenance therapy. If we think about resistance, so antimicrobial resistance, what you can see is that there have been cases of flucytosine resistance reported. And in this important case that was done by Tihana Buchanek's group, when she was in South Africa back in 2006, what they reported was that in patients who are on long-term maintenance therapy with fluconazole, you can see in patients who relapse, there is resistance in those strains in 67% of those patients. So drug resistance does seem to be a problem in this group. It's important to note this is prior to the induction recommendation. So this is patients who were solely treated with fluconazole. So it is an issue. If we think about sort of resistance mechanisms, we had a really nice analysis yesterday, both of the SNPs that are typically associated with fluconazole resistance in Candida albicans. So in this organism, you'll often find SNPs in ERG11, which is the target of fluconazole. Similarly, if you look at Aspergillus, you can see that there are often SNPs in CYP251A, which is the target of the drug triazole. Interestingly, I've told you that resistance happens in 67% of patients who relapse, but if you look for SNPs in those patients, you don't find them. Actually, SNPs in, for example, ERG11 are rare. So given that, this definite association with resistance that we're seeing in these relapsed patients, how can we bring these two ideas together? What is going on here? What's biologically going on here? All right, so this is work that was done by Junquan Cheng's lab, where they were investigating this problem and trying to understand what might be going on molecularly that's driving this resistance. And they brought up this term, it's called Hetero Resistance, which is the transient gain and loss of resistance to drugs. So I'm showing you just a couple of the experiments that they did. It's a really great paper. I recommend reading it. It's got a lot of cute molecular biology. But basically what they showed was that if you grow a clinical strain, H99, in the presence of fluconazole for a number of days, eventually you'll get resistance. And here's this is 64 micrograms per mil. So this is the break point, which she calls the MIC. So after continuous fluconazole exposure, you can see you've lost the zone of inhibition. If you then remove the drug and you grow the cells in the absence of the drug for even 16 days or 26 days, you can see the loss of this resistance. So it's transient, right? And they've termed this transient gain and loss hetero resistance. And when they looked at these cells, what they noted was that if you look at the double resistant strains, so these are plots of DNA density. So what you're looking at is if there's a line just across the middle, then you basically have one copy of your chromosome. If you've got two, then you have a duplication of that chromosome. So you're looking at aneuploidy here. So if you look in the highly resistant strain, you can see there's an aneuploidy both on chromosome 4 and on chromosome 1. And as you lose that resistance, as it's transiently lost, now you've lost that duplication on chromosome 1, and again, over time, lost that duplication of chromosome 4. So in the presence of this drug, what they're showing in vitro is that there's this rise in aneuploidy. And why is that important? It's not simply that aneuploidy converts drug resistance, right? It's the genes that are on that chromosome that are associated with resisting drug that is driving that resistance. So if you look on chromosome 1, there are two genes involved in drug resistance on the chromosome 1, ERG11, as we've already discussed with Candida albicans, and also this azole transporter, AFR1. And when you, basically what happens is when you have an aneuploidy on chromosome 1, now you have extra copies of these genes, you have extra copies of the target of the drug, extra ability to export the drug. And that's underlying, they've shown underlying this resistance. And they did a really cute series of experiments, basically where they moved ERG11 off chromosome 1 and showed that the chromosome that ERG11 is on will be the one that becomes aneuploidy. So if you put it on chromosome 3, now you see aneuploidy on chromosome 3. So that's tracking with this. All right, so that's in vitro. What about in vivo? So here they've done the same experiment basically in mice, and they've treated their mice. So the black is the untreated. The gray bars are the treated mice. And you can see that with fluconazole, there's fewer CFUs, so it does appear to be treating the mice, right? This mouse didn't survive to the end of the assay, whereas this mouse did. But again, if you look at ploidy in these cells, what you see is that before treatment, in the untreated cells, you've got one copy, and in the presence of fluconazole, now you've got aneuploidy. You've got upregulation of chromosome 1. So we're saying that aneuploidy in this organism is underlying drug resistance both in vitro and in vivo. Okay, so now we've got this sort of dual mechanism for achieving this goal, right? You can either become resistant to drugs through changing the target of the drug, right, through SNPs, or you can become resistant to the drug through the upregulation of that target. The important thing to note is that SNPs are genetic, so they occur in the DNA, and they're heritable. They're passed through the generations. Aneuploidy is epigenetic, if you'll let me use that term here, and it's transient. It's lost over time. So what that means is that if you're monitoring clinical samples and collections for resistance, you're going to miss it. It's very transient. I showed you between just 16 days is enough to lose that. And we see that reflected in the literature. So for example, Michael Fowler did a study in 2015 where he looked at samples from 2010 to 2012, and what he reported was no cases of azole resistance in Asia and Western Pacific. But that does not jive with what Tihana showed, whereas if you looked directly after patient collection, so these were samples that were collected and then analyzed at the site of collection. And she reported 67% of relapse cases have fluconazole. So what's underlying that, I'm arguing, is this difference in the mechanism by which this organism is acquiring resistance. It's not genetic, it's epigenetic and transient. Okay, so I've told you that this is happening in vitro and in vivo, but if you look at isolates of Cryptococcus neoformans from the environments, what you'll see is that actually aneuploidy is very rare. This is an organism that's really good at staying haploid. So here is a number of strains that have been collected from environmental and clinical isolates, and in all of these strains, what you can see is that, so they had about, the vast majority of them were haploid. They did find a few that were diploid, and a good number that were diploid here in these two serotypes. But these are all hybrids, they're AD hybrids. They're not, true. They're probably the result of fusion rather than through the aneuploid mechanism that we would see in the patient. And even if you look at these ones that are coming across as maybe serotype D, 100%, there's still 2 AD hybrids in there. It's only in the serotype A that you have 100% of the diploid strains, which is only 3 out of the 100 strains that they analyzed here being true diploid AAs. And these still may be AA fusion, it's difficult to tell. If you look at the DNA content, so these are 10 independent spores that I dissected, grew over time, and what you can see is this is a plot of ploidy, basically DNA content. And what you can see is that every single one of these strains has exactly the same DNA content. They're all haploid. So aneuploidy appears to be arising infrequently in nature, so why do we see it in the patients at such high levels? Okay, so given these differences in mechanisms, the answer to this question, I'm arguing, is that it's because Cryptococcus can induce aneuploidy in response, specifically in response to the host environment, and that is driven by this morphotype called the Titan cell. So this is a Titan cell. This is a section of a mouse lung that I took. You can see the Titan cell here, and I'm sure you all have seen these in histology sections as well, because we do see these in patients. So here's a regular yeast cell, and here's a Titan cell. It's much larger. It's got a very distinct cell wall, which you can see here. I'll talk about that in a second. And you can see that they're just growing in the lungs, simply the same way through proliferation that the yeast cells would grow. If you look at Titan cells under the microscope, there are some distinct differences between the haploid yeast and the Titan cell. First, I think the most striking is the size, obviously. So here's a haploid yeast. They're typically 5 micron. They can get up to about 9 or 10. But Titan cells, the definition of these cells is larger than 11 microns. So this one's 20. They can get up to 100 micron. They can be very large. They also have a very distinct cell wall. So you can see here it's a much thicker cell wall compared to the cell wall of the haploid yeast. And then finally, they have a distinct capsule. And there's been some nice work coming out of the Nielsen lab analyzing the differences in the capsule, Kirsten Nielsen's lab, if you want to read more about that. But it's, instead of being this nice fluffy capsule, it's this very, very dense capsule. The other really important thing to know about Titan cells is that they're polyploid. All right? So you go from being a haploid yeast to being a highly polyploid Titan cell. And this is DNA being stained here. And then the really puzzling thing about these cells from my perspective, the fascinating things about these cells, is that they don't go from being haploid to diploid to haploid again. They don't divide their DNA equally. They actually undergo asymmetric division of their DNA. So these highly polyploid 4N, 6N, 8N, or sorry, 4N, 8N, 16N cells can then go back to being haploid again. That's a very unusual property in life, being able to do that asymmetric DNA division. So you're seeing that here. So here's a daughter cell from a Titan cell, and here's another daughter cell from a Titan cell in the background with the lower DNA content. And I'll just show you that here. So what you're looking at here is a plot of cell size versus DNA content. So here's haploid in blue, the diploid cells in red. And if you sort by cell size, so these are the ones that are basically the Titans, they're greater than 11 micron. You can see that this highly polyploid population and then this very different population of spores with lots of different ploidies. So these are an inducible in vivo path to aneuploidy. So how do we study these? Well, if you look at the lineages, so if you do an induction where you take the yeast cells and you induce them, for example, putting them in mouse lung and then doing a lavage and then collecting those Titan cells and then dissecting those daughters that are emerging from the Titan cells and analyzing those populations. So these, I'm going to show you data here from the same mother, individual daughters that have come off, and then we've grown those into colonies. What you can see is that these daughter cells exhibit heterogeneous drug resistance. This is not an MIC plot. I'm simply showing you the ability of these cells to grow in the presence of YNB or when there's a sub-inhibitory concentration of fluconazole. So here's our haploid and our diploid, and what you can see is a spread of ability to grow better in the presence of the drug compared to the host, and the parental strain. And again, these are all coming from exactly the same parent. So just a simple aneuploidy that's being generated from a single parent. Now we're going from a clonal population that behaves very similarly to itself to a lot of heterogeneity. And if you look at the ploidy of those cells, what you can see is that some of them are haploid, some of them are aneuploid, some of them are diploid. So there's a big spread of chromosomal content in these cells. And again, this is transient. So this plot I looked at 36 hours after dissection, as close to after dissection as I could. And then I went away and did a bunch of other experiments and taught my students for a while. And then I went back to repeat the experiment. And now you can see that we've lost this drug resistance. So now it's transient. Like even after 1 month, we've gone from being this heterogeneous population to basically being all sensitive to the drug. All right, so In addition to the daughters being really important for generating population diversity, for generating drug resistance, the mothers themselves are very important reservoirs for infection in the host. Because of this large cell wall, they have increased drug and stress resistance themselves. They are very, very large, so much larger than macrophages, which means they are difficult to phagocytose and clear. We also see that when the Titan cells are present, there's increased eosinophilia in the lung. It's associated with a non-protective Th2 response. And we also see increased dissemination to the brain in the presence of Titan cells. So this is really important cell type. It's important for drug resistance. But the mechanisms underlying Titan cell formation are largely unknown. So this is where my lab comes in. What we're interested in understanding is how are you getting from these haploid yeast to these highly polyploid Titan cells, and then how are we getting from these polyploid Titan cells back to these haploid yeast again? So data I'm going to show you now is a few molecular slides. It's not going to be too bad, I hope. But what we've shown previously is that regulators of yeast morphogenesis are really important for the things that we care about in Titan cells, so cell size, cell ploidy. If you look at cells that are defective in RAS function, and you probably all know RAS is an oncogene, it's the ROGTPase is responsible for polarized growth and a number of other features in Cryptococcus. If you look at cells that are deficient in RAS, they basically can't form Titan cells. They have altered cell size, altered ploidy, which I'll show you in a minute, and they are not able to proliferate as Titans. We've shown also that if you lose CDC42, now you're defective in Titan cell formation, and that's published. I won't talk about that. What I want to focus on today is this protein called RAC, which is also a ROGTPase. And I'll just tell you a little bit about the ploidy story here between RAS and RAC. Now, if we look at, so the RAS mutant was made back in probably 1997 or so. It's an old Cryptococcus mutant, and we thought we knew basically everything about it. But if we look at the ploidy of this organism, so we published in 2013 when I was in Andy Alspa's lab, if you look at the ploidy of multiple different independently generated RAS strains, rather than being haploid, and here's our diploid control, the RAS mutant is diploid. And maybe that's just because there's something wrong with the strain. You know, you would argue, oh, well, this is an old strain, it's got some kind of molecular problem. But actually, if you induce RAS activity, now you're able to restore haploidy to these cells. So that's suggesting this is actually not something fundamentally weird about this strain. It's actually down to the regulation of RAS activity that's driving this. So we asked whether we could restore this by restoring activity of downstream targets of RAS. And what you can see here is that when you overexpress RAC1, now again, you're able to restore haploidy to these cells. So RAS is important for cell size. It's also really important for regulating genome content, which is one of the major features of Titan cells. So my lab is focusing on trying to understand how exactly RAC is acting. Now, I just told you RAC is the ROGTPase, it's acting downstream of RAS, but you also probably know RAC in the context of NADPH oxidases. And I'm sure you all know about NADPH oxidases. This is the complex that's responsible for reactive oxygen species burst that's generated by phagocytic cells. Fungi also have these complexes. And they act, rather than being a multi-protein complex, they basically have three elements. They have an NADPH oxidase, they have a regulatory protein, and they have RAC. And they act through the generation of reactive oxygen species, which we think now acts as signaling molecules to mediate functions in the cell. So the work that my lab has been doing for the last year and a half has been identifying the proteins that are involved in this reactive oxygen species complex. We now know that Cryptococcus encodes 2 NADPH oxidases, a NOx regulator, 2 RAC proteins. So this complex is intact in this organism. And if we look at the Titan-related phenotypes in these cells, which you can see is, again, here's my haploid cells. If we look at cells that have come out of these mutants, so here's a little bit slightly different looking at the DNA content. The haploid is always in blue and the diploid is always in red. So here's our RAS being diploid. If you lose NADPH oxidase activity, now you're no longer able to regulate your genome content. Now you start getting altered cells coming out with sort of abnormal aneuploidy, either diploidy or just like not very clean genomes. And similarly, if you lose RAC and NADPH, the regulator activity, you get this spread in ploidy regulation. All right, so what I hope I've managed to convince you is that these genes are involved in regulating these Titan-related phenotypes, and that's going to be able to give us insight into understanding the molecular mechanisms of Titanization in this organism. And I just want to remind you again that Cryptococcus neoformans cells have these Titan cells that are an inducible in vivo path to aneuploidy, and that's really important for underlying the drug resistance that we're seeing in patients. Now, Cryptococcus is not the only organism that's doing this. Aneuploidy is important in drug resistance in a number of other systems, including Candida albicans. So Judy Berman's lab has shown nicely that if you expose Candida albicans cells to fluconazole, there's an alternate mechanism for these cells to reach aneuploidy, which is the formation of trimeras, or multiple nuclei, which then lead to isochromosome formation and aneuploidy. And if you'd like to know more, then I suggest reading this wonderful review by Jingquan Chung and Yui Chang, which is called Aneuploidy and Drug Resistance in Pathogenic Fungi. It came out in 2012, but it's still very relevant. I will stop there, and thank you for listening. And I'd just like to point out, if you are interested in following more about our lab, you can find us online at the MRC Center CMM. And I'm also online at Blue Lab on Twitter. I'm always happy to talk about science. Thank you very much.